Okay, welcome to review session number 2 on photosynthesis, pairing up with our reactions that we just did in the mitochondria. We're going to do similar types of reactions in the plant. I guess I should kind of back up and add an extra slide here. So last reaction or last review that you saw, we saw this reaction taking place. We talked about all the components and where they're used, right? So this is first used in glycolysis. This was the final electron acceptor, right, which eventually turned into water. And this CO2 came from the original sugar molecule, so that was released from the Krebs cycle. When we talk about photosynthesis, it's basically the exact reverse of this reaction that you were seeing in cellular respiration. Obviously, we got to put in the balanced stoichiometry in me needs to have consistency. So now with photosynthesis, we had the exact opposite of this reaction. So we're producing the reactants from cellular respiration become the products of photosynthesis. So the whole goal here is to make high energy chemical bonds in sugar from taking light energy. So on this arrow here of the reaction, we're going to use light to drive this reaction. And so we're going to need to somehow take air and water to make sugar. So this is the most confusing part, or the most amazing part to me of photosynthesis, is we're taking something that is gas and we're turning into something like sugar, right? So that often becomes a quandary or one of those deep thinking type of things. We're taking gas of CO2 and we're turning it into sugar somehow. And it all starts off with the light reactions. Now, these light reactions are going to take place in our main organelle of plants, which is the chloroplast, specifically in the thylakoid membrane. So this will come back. I'll show it to you again. So the thylakoid membrane is where we're going to see this. So just like we saw a lot of the main reactions for cellular respiration, especially the electron transport chains found in the cristae, we're finding an inner membrane within the chloroplast is where we're going to see the light reactions taking place. All right, so there is a main molecule that is going to do a lot of work here. It has to be present in order for the light reaction to occur, and that is this molecule right here, which is CLA, also known as chlorophyll A. And chlorophyll A is the only pigment that can actually pass electrons to the primary electron acceptor. So I'm going to draw in our electron. All right, so that's going to represent my electron now. I'll put a little, I should put a little E in it, but now the E is not going to move because I put the marker down. So that electron, if you remember from our cellular respiration, when it came downhill, the final electron acceptor was oxygen and it turned into water. So that means that the electron in water is a very low energy electron because it did all the work of all those protein pumps along the membrane of the cristae. So now we have a low energy electron that's found in water. So if you see in this diagram, yep, see, the E didn't come with. So this electron is in water molecule right now. Chlorophyll A has electrons in it. So if chlorophyll A's low energy electron, right, so we'll give chlorophyll A one right here. Chlorophyll A's low energy electron gets excited, right? So that's what you're showing. You're seeing light coming in here, right? So light's coming in. It's going to excite that electron. Now that electron's high energy. It pops up, and the primary electron acceptor has a baseball mitt, and it grabs that electron. Well, now if you think about it, chlorophyll A has got a dilemma. It doesn't have an electron. So water has to be split to fill the void of that chlorophyll molecule. Now, you don't have to understand the details of all the components of how that happens. Just realize anytime you see any complex work found in any system, there's probably an enzyme there. So there is an enzyme there that is actually taking water and helping split it so that we produce an electron for chlorophyll A. The byproduct then, as you can see, is oxygen. So this is where we're using water and where we're producing oxygen when we're talking about that original formula that I just showed you on the last slide. So we're talking the splitting of water is going to give the electron back to chlorophyll A that just gave its electron up because light excited it. Now the primary electron acceptor has the electron, and you can see, oops, recognize it as an O. We don't want to. So we're going to let that electron roll downhill just like we saw in the cristae. So what you're seeing is these, I don't know, what are these teal blobs here? These teal blobs are like the electron transport chain that you just saw in the last chapter, right? So they are the protein pumps that are found in the thylakoid membrane. So what are we talking about? We're talking about transferring H pluses from one location to another against their gradient. Well, just like we saw with chemiosmosis and cellular respiration, if we pump all these H pluses, for example, like this, right? So here's the thylakoid, right? And here's outside in the stroma. We're pumping all these H pluses into the thylakoid. Well, now if we let them go back down through that selective doorway that we really want them to go through, we can make ATP. So as you can see, the electron rolls downhill and we make this ATP. Right, so that was all because of the same principle that we did with chemiosmosis in the mitochondria. Now you can see there's another chlorophyll A. Yes, it's P680 and P700. All that does is it's a slightly different wavelength that these two chlorophyll molecules are absorbing at. Now, once again, this is still chlorophyll A, right? And the funky thing is, when they first were trying to figure all this stuff out, they found chlorophyll A from the P700 category first, so hence that's why that's photosystem 1. And then they're like, oh, wait a second, we found another photosystem, so we better name that number 2. So that's why photosystem 2 comes before photosystem 1. Thank you, biologists. So chlorophyll A now has light coming in as well, and chlorophyll A's has an electron. Right, so here's chlorophyll A's electron, and that chlorophyll A electron gets excited and jumps up to another primary acceptor. Well, just like our issue with P680, if it loses an electron, we have an issue. Nice thing is, we just use an electron going downhill. It's very weak now, and it fills the void of chlorophyll A. Right, so now we have both chlorophyll molecules here and here that are completely happy and ready for the next cycle of any light coming in. And now that electron got captured by another primary electron acceptor, we're going to go down a small transport chain, but we're not really going to make ATP. And what we're going to do is we're going to dump it into a molecule that can accept electrons, much like NAD+ or FAD. So when it gets that electron, it can store it, and it becomes NADPH. The way I always remember it, P for plants. So you're going to see things on the test. Oh, NADH, ATP, NADPH, right? FAD2, H2. So they're going to say, oh, which one's found in plants? Well, P for plants, that's your electron carrier in this system. Now, this is non-cyclical photophosphorylation. Can't even fit it on the entire screen. You know what I'm talking about. So, non-cyclical. It's not going in a circle. And if you look, it kind of makes this N type of shape, right? So that's the way to remember it. So this is non-cyclical photophosphorylation. This is where we're going to actually use water and produce oxygen. And the two products that are big that we make are ATP and NADPH. The counterpart to non-cyclical photophosphorylation is cyclical photophosphorylation. Now, remember, we've already talked about a couple of phosphorylations from the last unit. So here, once again, you're seeing that N shape. This is non-cyclical photophosphorylation. So we use water in this one, and we produce oxygen. We make ATP, and we make NADPH. The other one, cyclical photophosphorylation, that electron that photosystem 1 had with the chlorophyll A molecule, that gets excited by the sunlight's energy. It passes up to the primary electron acceptor. It comes downhill like an electron transport chain, but instead of going to another photosystem, it fills its own void. So now we have a chlorophyll A molecule that keeps filling its own void. The difference is no water is used here because we don't need any new electrons. But because the electron doesn't get stored in a new molecule like NADPH, we only make ATP in this one. So you ask yourself, well, why don't we do just this one? Well, we do need NADPH when we get to the Calvin cycle. So depending on the plant's needs, the plant can actually shift between its needs that that it has and the wants that needs and wants, I guess, that it needs, has, what? You know what I'm talking about. So we basically need more ATP, we're going to do more cyclical. If we need more NADPH, we're going to do more non-cyclical. But the key is that these are both photophosphorylation processes. So in review, we have substrate level, right? If you remember that, that was in glycolysis and a little bit in Krebs cycle, or substrate level, phosphorylation, spelled off the side of the sheet here. And that was done by the kinase enzyme. And then we had oxidative phosphorylation. And this was done with the chemiosmotic process that we see in the electron transport chain. Remember, we oxidized basically food, glucose, and we ended up making NADHs and FADH2s, and that powered the electron transport chain. And now we see photophosphorylation. And photo, meaning light, we're using light energy to phosphorylate and produce ATP, right? But we're still using an electron transport chain. So there's quite the comparison between the two, and we're going to look at those in a little bit. So coming back to our organelle of interest, which is the chloroplast, this is a double membrane organelle, just like our mitochondria. So we've got the outer membrane, and then we've got this inner membrane. Good choice of colors, green on green. Good job. So this is thylakoid membrane. That thylakoid membrane is the inner membrane. So it'd be similar to the cristae/matrix area that you see in the mitochondria. So we have a stroma, which is the inside but not past the thylakoid. Right, so you're seeing it here. Right? And then we also have the thylakoid. Now, comparison-wise, let's look at this. We see the mitochondria. This image is straight out of your textbook. It's a little different coloring. I think they have peach and salmon colors for you as reference. But you can see we're going to use an electron transport chain, right? What powers the electron transport chain? Let's think about this. There's two different ways. If we're talking mitochondria, what powers the electron transport chain is going to be our NADH, right? If we're talking about powering the electron transport chain for plants, it's going to be the sun, right? Regardless, we're going to allow electrons to move through proteins and pump H pluses into a specific area. And then we're going to allow them to do a facilitated diffusion process back through ATP synthase to produce ATP. Now, comparison-wise, we have matrix, inner membrane space, thylakoid, stroma. They're very much the same process, except we're pumping H pluses into this inner membrane space, and then they're rolling back into the matrix, right, through the ATP synthase. Here we pump them into the thylakoid, and they roll back into the stroma. So make sure you understand the comparison here, right? And this whole process that we're talking about comparison is chemiosmosis, right? Which is basically the pairing of the electron transport chain to the ATP synthase. I'm hurting myself here trying to fit it on this page, right? So the pairing of electron transport chain to the ATP synthase, that's chemiosmosis, right? So don't get confused. There's a lot of words that come up that they sound very similar, like proton gradient, proton motive force, chemiosmosis. Make sure you understand the comparisons between those things. And if you have any questions, please email or come and talk to your teacher. All right, so we have just talked about the light reactions, right? And those happen, like I said, in the membrane of the thylakoid, these stacks of pancakes, these green stacks of pancakes that are found inside the chloroplast. They produce, right, NADPH and ATP, high energy molecules that we're going to use to power the Calvin cycle. And the Calvin cycle is the dark reaction. And the dark reaction is found in the stroma, right? So the dark reaction is the fixing, right? We're going to take in CO2 and somehow we're going to make sugar molecules. The way we do this is by using energy. The energy that we've taken from the sun and we've put into molecules that are highly energetic, right? So keep this in mind, right? Here it's showing we're using water. Is water used in cyclical or non-cyclical? Question you need to ask yourself, and this question is on the test. The answer is non-cyclical, right? We produce oxygen that way as well. If we're talking about cyclical, we really don't use any extra reactants other than light, right, and any other small molecule like ADP that we're going to turn into ATP. All right, so let's turn our attention to the Calvin cycle, which is the dark reaction. So the dark reaction, Calvin cycle, once again, happening in the stroma, right? Our inputs in here, CO2, right? And out comes this G3P molecule. G3P, if you do remember any of this complexity, is found actually in the glycolysis component. So when we were talking about cellular respiration, we were talking about sugars getting split up. A lot of the sugar components are very similar, right? And so there's a very common component, G3P, glycerol aldehyde 3-phosphate, is going to be found in our glycolysis as well. So it's going to take 2 of these G3Ps to make a C6H12O6. So what am I telling you is, in order to make a glucose molecule, you're going to have to turn the Calvin cycle 2 times, which means we're taking in not 3 CO2s, but 6 CO2s. There's a big enzyme that you have to know with photosynthesis, and that's Ribisco. Ribisco is the fixing enzyme. So it's going to fix CO2 into our system. So we have a molecule that's already there, just like we saw in the Krebs cycle. There's a molecule already there. So RUBP is going to be added to the CO2 via this enzyme, and in turn you get a new molecule out. Notice with the Krebs cycle we made high energy molecules, right? But here we're going to use high energy molecules to do work. We're going to build something, right? We're going to put energy in, so it's not going to be an exergonic, it's going to be an endergonic reaction. So we're going to use ATP and NADPH to actually reduce our sugar. Remember, Leo goes GR, G, E, R. Gain of electrons is reduction. So we're taking electrons out of NADPH, and we're going to put them into the sugar molecule that's there, and we're going to make it a molecule that's even more highly energetic. And that molecule then comes out as a sugar molecule, and we continue through, and we end up having the end product is going to be a reactant for another turn of the cycle. Those are the details that you need to know with the Calvin cycle. I feel like it's a little less detailed than the Krebs cycle. You basically have to know that what enzyme is going to fix in the CO2, what's coming in, the fact that there's a molecule that's already there, right? And in turn, we're going to use ATPs and NADPHs in order to get products out of the cycle so that we can use them and store them as sugar to feed the entire world, basically, right? If we don't have this process happening, there will be no extra energy for life processes. All right, comparison now. That was considered C3 production, because if we go back, right, the carbon that we produced was 3 carbons long, right? The problem is Ribisco. Ribisco is this enzyme that has issues. If we're in an area where it's very dry, on the bottom half of leaves, right, a lot of times it's on the bottom half, so on the underside of leaves, there's little pore openings that kind of look like this. They have these guard cells that are called stomata. And those stomata open and close in order to have gas exchange. So plants want to take in, not O2, plants want to take in CO2, and they want to get rid of O2. But as they release O2, they also release H2O. I don't know if you've ever experienced this, where you lay a piece of tarp or a piece of plastic on the grass in your yard during the middle of summer, and you go to pick up that piece of grass or that piece of plastic, and there's all kinds of water on the bottom side of that plastic. That's because the plants underneath were transpiring. They were releasing some of their water as they were trying to breathe. Now, plants need water, right? We've already talked about how the water in their vacuoles helps them stand upright, especially plants that don't have a lot of secondary cellulose in their cell walls, like trees. They're going to have water in there. So your petunias, they droop over because they're lacking water. You give them water and they stand back upright. So if a plant is in a dry area and is trying to get in CO2 so they can fix it, make sugars, and get rid of O2, which is basically their toxin, they're going to be releasing water as well. Well, if it loses too much water, the plant can die. So the plant has to close its stomata sometimes. Well, if it closes its stomata, what starts building up? O2. The issue is Rubisco loves O2, right? It actually loves O2 more than it loves CO2. So what does it start doing? Instead of fixing CO2 like it's supposed to, it fixes O2. And we end up using energy instead of making high energy molecules, and that's called photorespiration. If the plant photorespires, we're not making sugar. We're actually using the energy and wasting it. So along long-term time, right, in drier areas, we see two different evolutionary processes that have developed, the C4 and the CAM plant, and you have to know them both. They both have a new enzyme that's found. It's called Pep Carboxylase. Pep Carboxylase can actually fix in CO2 a different way. So Pep Carboxylase has no love for O2. It doesn't care if O2 is there. It only fixes CO2. So in a C4 plant, what we're doing is we're seeing a separation. I know you can barely see this. I can barely see it, and I'm standing next to the board. A difference in cells, right? So what's going to happen is the CO2 is going to be fixed into this 4-carbon molecule, C4, in the mesophyll cells. You don't have to know the details about what mesophyll means, but it basically is a different cell than where we're going to do the actual Calvin cycle, which we focused on already. So the bundle sheet cells is where the Calvin cycle, or the dark reaction, is going to occur. So we're going to fix up all the carbon into this molecule, C4. We take it down to the bundle sheet cells, and we're going to tear off a CO2 and give it to the Calvin cycle, where Rubisco still sits. Right? So the idea is we're going to store CO2 in little canisters called a C4, and we're going to release that CO2 so that Rubisco doesn't accidentally do this photorespiration process. So it's a way of storing up CO2 in a special spot and giving it to the Calvin cycle to be done the correct way, right? The CAM plant does very much the same thing, but instead of having it open and close during the day, what they do is they open up their stomates during the night. They fix in all the CO2 during the nighttime, so they build up all this CO2 when their stomates are open, and then during the day when they close the stomates, they start releasing all the CO2 to the Calvin cycle. So this isn't a separation of cells, this is a separation of night and day when we're actually doing this fixation process. So once again, evolutionary adaptation, C4 versus CAM. CAM, here's the way I always remember it, CAM, AM, PM, right? So night and day. It stands for crustacean acid metabolism, if you really wanted to know. But it's the separation of AM and PM when we're going to fix these carbon molecules to be released. So think of it as, right, we have a canister in this idea. We're going to put the CO2 into the canister, right? And then we're going to transport this canister to the Calvin cycle and release that CO2 to the Calvin cycle. And then we'll end up making the sugars that we need to do, right? And as you can see, we see this in very dry environments, tropical environments like pineapple and sugarcane that can't be grown in Illinois. They have to be grown in a much more humid environment that has constant sunlight. So we're going to see those adaptations in those different places. I believe this concludes the entire process of photosynthesis. If you do have questions, please feel free to ask. I think I've hit all the key points here. Yep, there's no more slides. So I wish you well, good luck, and keep studying.